The research program is really about developing a drug targeting system to deliver bioactive molecules to a specific organelle within human cells, and that organelle is called the lysosome. And the lysosome is basically the waste bin of the cell, and it's where products from cellular metabolism go to get degraded after they've served their lifetime. And the specific medical application of our research relates to a series of about 60 or 70 really debilitating fatal diseases called the lysosomal storage disorders. Now, these are a series of diseases which are genetically inherited. They're classified globally as rare, but there are a couple of hundred sufferers in New Zealand. Each of these diseases relates to a lack of a functional degradative enzyme in the lysosome. Children are born normally. As they go through infancy, what happens is these products that need to be degraded build up within the lysosomes of their cells. So they start to develop symptoms because an enzyme which does the degradation process is missing, is not functional. So they develop symptoms, and then there's a whole series of pathologies which is different relating to the different diseases. Untreated, they live for a varying period of time, they go blind, they become immobile, and then without treatment they all die. So there are no cures for any of these diseases at the moment. One of the potential treatments for these diseases is something called enzyme replacement therapy. And in enzyme replacement therapy, what you do is that you produce the missing enzyme that these children lack in a cellular expression system in the laboratory. And then you inject this missing enzyme into the patients. And this is a regular treatment, it's not a cure, and has to carry on for the lifetime of the patient. Now, the big problem is that there's a huge difference between injecting an enzyme into somebody's bloodstream with a syringe and that enzyme finding its way through the blood system into the particular cell type and then getting through inside the cell and then getting to the lysosome where it needs to be active. The key aspect of our research is to develop organelle targeting so that we can specifically deliver enzymes to not only into cells but to the lysosome so they get to where they need to do their job. Now, the question is, how on earth do you think you're going to do that? What we've decided to do is actually to copy or hijack the natural targeting mechanism by which the cell delivers these enzymes to the lysosome, because of course normally the cell makes lots and lots of enzymes, so there must be some specific mechanism which says send this enzyme to the lysosome where it needs to do its job. There is a natural system for delivering enzymes produced within the cell to the lysosome, and that uses a specific tag, and that tag is called mannose-6-phosphate. And our project is all about producing enzymes or other species, other drugs that we want to deliver to the lysosome by specifically attaching this tag called mannose-6-phosphate to those cells. The opportunities of this technology that we're developing are really manifold, not only because there are more than 70 different lysosomal storage disorders, each of which you could begin to treat using this technology, but there are also a whole other array of disease states which relate to lysosomal dysfunction. There's a disease called Stargardt's disease, which is untreatable and which causes blindness, that we could also treat by delivering different sorts of biomolecules to the lysosome.